Hi everyone, my name is Vikram. I'm a postdoc in the lab. I've been here about 2 years now. My background is in wet lab biochemistry. I did my PhD at the University of Toronto studying protein folding experimentally. So when I got here, there was a bit of a culture shock trying to figure out how to use these tools and how they work on the inside. And so I thought it would be a good idea to have a talk in plain language for new Rosetta users explaining the core Rosetta algorithms without a lot of computational gobbledygook. There is going to be a little bit of math in this talk, but it's mainly going to be math described in plain English. There is not going to be any computer code in this talk. So, getting right into it. Broadly speaking, what does Rosetta do? At its core, Rosetta is a bunch of algorithms and infrastructure for keeping track of protein structures or any heteropolymer, just chains of residues, their kinematic relationships and that sort of thing. These feed into algorithms for manipulating conformation or sequence, for searching sequence space or conformation space, and algorithms for scoring the favorability of a sequence of a particular conformation given a particular sequence. So you're going to put the protein into some sort of conformation, and then you're going to score it and ask, is this a good conformation? So there are a bunch of concepts that are Rosetta specific that we need to cover. One of which is the fold tree. So I told you that Rosetta handles chains of residues, and you can do things to those chains. You can torque around various torsion angles. That creates certain problems. And here's an example. Let's say you had a structure that had two chains. You had a target that you were designing for, and you had something that was binding to it. And let's say I arbitrarily decide that I want to rotate around a bond here in the middle of this chain. There are two things that could happen. Either this part of the protein could move so that I get something like this, or alternatively, this part of the protein could move and start clashing with my target. And if I just said torque around that chain, it wouldn't be clear what would happen. So Rosetta uses something called the fold tree to define a hierarchy of conformational inheritance for residues. Every single residue in your structure has a parent residue which will remain fixed if you rotate about the bond between that residue and its parent. And for a single chain, usually you'll have some sort of pattern like this, where each residue is a child of a previous one. There's also something called jumps in Rosetta. Jumps are rigid body transformations, usually between two different chains. So if I have one chain and another chain, I can say the change in x, y, and z coordinates and the change in three rotational values to get from one to the other. Jumps are actually defined on the residue level. So the residue in this chain that is connected by a jump to a residue in this chain will inherit this residue's x, y, and z coordinates before the jump is applied. So if I were to rotate around a bond here, chain A would move, and also chain B would move because it's linked to a residue downstream of that bond that I'm rotating about. The inheritance pattern does not have to go in linear sequence. In the example here, the root of chain B is in the middle of the chain, and the termini are downstream of chain B. That can be useful in situations in which you have two things docked together, and you want the root to be something close to the binding interface rather than something that's far away from it. In Rosetta scripts, you can change a fold tree using the atom tree mover. There are fold tree files. All of this is in the documentation. I'm not going to get into the details of that, but it is sometimes worth thinking about the fold tree that you want for your particular problem. The fold tree that's good for one problem might not be the good fold tree for another problem. So fold trees just define the kinematic relationships between residues. Does that all make sense? Any questions? Moving on. What is conformation space? This is a concept that's going to underlie a lot of what we talk about today. Conformation space, I think we're all familiar with 2- and 3-dimensional spaces and vectors in 2- and 3-dimensional spaces. So a vector is a little arrow with magnitude and direction. If I say 5, 3, 7, that defines a vector in 3-dimensional space. And if I have a single atom, I can tell you 3 coordinates, the x, y, and z coordinates for that atom that define its position in 3-dimensional space. And you can think of those 3 coordinates as a vector in 3-dimensional space. If I have 2 atoms, I can tell you 2 vectors, x1, y1, and z1, and x2, y2, and z2, defining their coordinates in 3-dimensional space. All of this is very basic so far. But rather than telling you 2 vectors, I could also just tell you 1 vector in 6-dimensional space, x1, y1, z1, x2, y2, z2. So this system, this 2-atom system, any state of this system corresponds to a point in a higher dimensional space that has 6 dimensions. And this applies no matter how many atoms I have. I could have 5,000 atoms in a giant protein, and if I tell you the x, y, and z coordinates of all of those atoms as a giant 15,000 dimensional vector, I've defined a point in a 15,000 dimensional space corresponding to a particular conformation of that protein. Why is this useful? Well, one of the reasons it's useful is it feeds into a concept that you've probably all used and heard about, but might not have thought about in this way, called RMSD. If I want to compare 2 structures, I want to calculate some number corresponding to how similar or different those 2 structures are. And if I've got these 2 structures, structure 1 and structure 2, and I've got vectors corresponding to each of them, in that higher dimensional space, the length of the difference vector between them, that's the vector that joins them tip to tail, is given by this formula. You take the differences of all the coordinates, sum up the squares, and take the square root. And this is the RMSD formula. So distances in higher dimensional vector spaces correspond to differences or similarities between structures. The closer to get together 2 points are in the higher dimensional space, the more similar the corresponding structures are. Everything I've talked about so far has been with respect to Cartesian space. If I want to tell you the conformation of a protein, I have to list x, y, and z coordinates of all the atoms in the protein. For the most part, Rosetta doesn't work in Cartesian space. We simplify the problem because we can work in a lower dimensional space defined by dihedral angles, bond angles, and bond lengths. So dihedral angles are when you have 4 atoms, the angle about the bond connecting the middle 2. Bond angles are defined by 3 atoms. The squishers stretch like this. And bond lengths are defined by 2 atoms. But many of the same ideas apply. You can define a vector in a higher dimensional space consisting of all these dihedral angles, and bond lengths. The nice thing about this vector is going to be shorter than the corresponding vector if you defined everything in x, y, z coordinates. So we're working in a lower dimensional space than the full Cartesian space, which simplifies some calculations. Any questions so far? Okay. So this leads into the next question. What is the energy landscape? This is a term that we toss around all the time. So given a conformation space for a protein where every single point in this space, and remember this is a space with more than 3 dimensions, it's got zillions of dimensions, well, thousands of dimensions. Every point in this space corresponds to a conformation of the protein. We can define an energy function that assigns a real value to every point in that space. So if it's a good conformation, that value is very low. If it's a bad conformation with lots of clashes or poor interactions or lack of good interactions, that value is very high. I can't draw a 1,000-dimensional space, but I can draw a 2-dimensional space. So imagine that the points in 2 dimensions along the horizontal axes correspond to conformations of a protein. And I've added a third dimension here corresponding to the energy value that you get at each of those points. For real proteins, for natural proteins, proteins have kind of a funnel-shaped energy landscape where there is a unique energy minimum corresponding to the native state of a protein, and the overall shape of the landscape guides a protein to that minimum. So it doesn't have to exhaustively sample the space in order to find that minimum. It can partly find that minimum by rolling downhill, more or less. The other feature of protein energy landscapes, though, is that they are very, very rugged, even on a relatively small scale. You can move a protein just a little bit from a good conformation and find a bad conformation that's very high energy. So although there's an overall funnel-like shape, there are many, many local minima, which means that we can't find global minima for proteins. We can't find the global energy minimum, the native state, just by rolling downhill. We have to have cleverer algorithms that search that conformational space a little more intelligently to find that global minimum. Does this make sense? Okay. So back to Rosetta. If we're going to put the protein in a given conformation and score how good or bad we think that conformation is, we need to have a good approximation of the actual physics. We need a good scoring function that to some degree corresponds to reality, or we hope corresponds to reality. And Chris is going to jump in here and tell us all about how Rosetta's scoring function works and what the terms are in it.
Usually when people say score function in Rosetta E's, what they mean is some set of score terms, each score term capturing some facet of protein thermodynamics, all weighted together into some sum. Inside Rosetta codebase, there are hundreds of score terms, some of them more experimental than others, some are deprecated, some used to be used, some are no longer used. What I'm going to be talking about are just the score terms in what is, actually this should be Tolaris 2014 now exists, still the same. Set of basic score terms that's for, giving your kind of unbiased estimate of a new problem, you'll start with that and then go from there. So I'm just going to start out with, say, given an interacting set of residues. So in the score function in Rosetta, all energies are calculated between residues. Everything has to be what's known as pairwise decomposable. You hear about this a lot, and what that means is that the total energy is a sum of the residue pair energies. That means that if there's some unique feature of the energy that's defined by the precise orientation of 3 or more residues, that's not going to be in the standard score function because it takes too long to calculate. So the things in Rosetta are residues. There's atoms in those residues, and we account for those. And those things are parts of larger molecules. But the core idea of things are residues, even when it's a polymer. That's why even with ligands we consider a polymer of one. So the first one, so what I've done is the top is the actual physical interaction. In bold are the names in the score function. Like if you look at the bottom of a PDB that Rosetta spits out, there's going to be a list of all these score terms. And so this is all the score terms that correspond to this actual physical interaction. And then where appropriate in italics, I've put the kind of like Baker Lab jargon, what people mean when they say. So when people talk about, so if David just says the word attractive or he just says clash, what he's talking about is van der Waals energies. And van der Waals energies in Rosetta are represented by these tags in your score weights file. And so this is just Leonard-Jones, or this is van der Waals interactions where you have a couple of atoms. This is kind of my wishy-washy understanding of van der Waals. I think it's kind of complicated. You have a couple of atoms with electron clouds around them. You have random fluctuations in the probability distribution of those electrons, and that can induce polarization in another atom or another molecule. And so any atoms, no matter what their charge, if they get close enough, they'll have, even if they're neutrally charged, they'll have some attraction once they get close enough. But if they get too close, they push each other away because of the Pauli exclusion principle. One easy way to account for that is what's known as a Leonard-Jones potential. In molecular mechanics, this is really, really common, where you just have some repulsive term and then some attractive term, and it bottoms out at whatever we've like experimentally determined are the usual distances between those atoms and crystal structures. So we have that, and that whenever there's a clash, it's that term that blows up. So if you see that term is like 10 billion or whatever, it means atoms are on top of each other. So the next one is electrostatics. You hear people call this hack-elec too. That's old jargon carried over. What they mean is FA-elec. The FA in all these means full atom. One thing I'm not talking about right now is the centroid score function. There's a very low resolution score function for when we represent all the residues just as just all the residue side chains as big spheres. And so FA is from, so Rosetta originally was centroid only. Only later did it actually add all the side chain atoms. And so that's why everything is like denoted FA. So this is just charge-charge interactions. We just do this using Coulomb's law. Right there, good old Coulomb's law. Except for this guy. This is dielectric constant. And so when we're in water, which is a very highly dielectric medium, say we have positive charge over here, negative charge over here, water, which has a dipole itself, will orient to counteract that charge separation and create its own charge in the opposite direction and cancel out some of that dipole. I might be throwing these words around a little loosely. And so to represent that, if we just have a constant dielectric, like say air or something, or I don't know, insulation around a telephone wire, we can represent that just with a constant of that reduces the amount of charge between two interacting particles or two interacting surfaces. In Rosetta, what we do something really simple. This dielectric constant is just a function of the distance between the atoms, and it just goes up in this kind of sigmoid type shape. That's a really gross approximation because if two atoms are separated by a distance of, say, 10 angstroms, in water, there will be very little electrostatic interaction between them. But if they're separated by, say, 10 angstroms through the middle of the protein, that's very similar to 10 angstroms through vacuum. And so the interaction between those charges will be much stronger, but we count them the same in Rosetta just for reasons of efficiency. And this is where some of the more complicated models that are in Rosetta, called like Poisson-Boltzmann, you hear about EFON's PB solver, try to actually account for these differences in dielectric. So solvation, this is kind of the biggest elephant in the very big room that is all of the approximations in the score functions. So this is called FA sol, or solvation. So we don't generally, there's, now and again, like more and more hearsay about doing explicit water in Rosetta, like comes up and then it dies down, it comes up and dies down. So, maybe it will work someday. But right now we don't account for water explicitly in Rosetta. We just consider it some invisible continuum, and then we try to account for the interactions of water in this kind of rough way. So how this works is also known as the LK model. Lazaridis Karplus came up with the original one. You start out with a whole bunch of reference compounds, small organic molecules. Actually experimentally measure the free energy of solvation for all of those. And then from all of those, and looking at, say single atom substitutions in two related molecules, you can kind of backtrack and come up with what in a wishy-washy way you could call experimental delta G's of solvation for that single atom type. So say you have an amide nitrogen, like what on average is the free energy of solvation for a nitrogen when it's in an amide group? And then you can transfer those to like any amino acid or any ligand you want, and then just add them all together. Like if you were to fully immerse that atom in water, that's the free energy that you would gain or lose. And so each atom is a sphere of a certain size with kind of Gaussian density. So it's strongest in the middle and weaker going out. And inside that sphere is all of that free energy that you calculated from your reference state. And then any other atom, any other sphere that occludes that by some amount, the more volume it occludes, the more free energy of solvation you just lost, because all the hypothetical water atoms that say could be inside the sphere around this atom, they can't be there anymore. And so you just lost that percentage of your free energy. So it's just your reference minus some function of the occlusion times the occlusion volume. And so this is a solvent exclusion model where we're just looking at hypothetical solvent we're excluding, which is way different from most solvation models. So here, like I talked before about how water screens electrostatic interactions. So you could think of that reduction in electrostatic interactions as a solvent effect, but we don't count it in our solvent model because we already counted it in the Coulomb's law part. And so we don't want to count for this at all. But then all of the molecule water and water-water interactions, like conformational entropy, everything gets all mashed into one number, which we tried to calculate from this reference state. And we just subtract from that. So next one is H-bonds. So this is the score term in Rosetta that kind of sets it apart the most from your standard like molecular dynamics type score functions, force fields, if you're in that world. And so this is one of the things that makes Rosetta work. Like after this was mostly invented by Tanya Kortem and some other people, it seems like that's when a lot of things started to actually work. Because it's really hard to get correct hydrogen bond geometries just from Coulomb's law and Leonard-Jones potential. And so all you do is you go to the protein data bank where you have a whole bunch of high-resolution crystal structures, pull out a bunch of things that you've annotated already that to your eye look like hydrogen bonds. From those you can derive distances, relative torsions, and angles, just like Vikram was describing in kind of torsional conformational space. And then for each one of those observables, you can get probability distributions. And all you have to do, as we know, because of Boltzmann, is take the log of a probability distribution, and you have some value that's kind of like an energy. You can scale that by whatever temperature you want. One thing we do, as far as scaling that probability distribution to energy type calculation, is we have environment-dependent H-bonds. The thermodynamic justification for why hydrogen bonds are counted as weaker on the surface of the protein is, I'm not sure. I think this is mostly just a heuristically motivated thing of.
It's competition with water.
Yeah.
You have less of an energetic benefit to what hydrogen bond on the surface of the protein.
Right. Yeah, that makes sense. Kind of. Yeah. So if you're on the surface of a protein, hydrogen bonds are weaker because making a salt bridge on the surface doesn't matter because they're all sticking out into solvent, moving around. So we have that. Very similar to that, as far as implementation, is the frozen computer is the disulfide bond term. So we basically do the same thing as we go to the PDB, pull out a whole bunch of disulfide bonds, derive the same kind of geometric observables, and then invert those into an energy somehow, using basically the same idea. Implementation is slightly different. And last but certainly not least is torsional strain. So one thing about 1,4 neighbors, like what that means is, say you got 4 atoms, like 1, 2, 4. So these are chemical bonds. Any atoms that are connected by 4 or fewer chemical bonds are not counted. None of the intermolecular, interatomic energies I've been talking about are counted in these. These are called like 1,4 interactions. The reason for that is because at this distance, in this scale, when you're sharing orbitals, when you have covalent bonds, quantum mechanical effects start to outweigh some of these more classical effects. And the approximations we're making, even considering this classical system in the first place, no longer makes sense. And it makes a whole lot more sense to just look at our downstream observables that we care about, the actual geometric arrangement of these atoms, and just derive distributions from those and implement those than trying to recreate it using all of these classical equations we've come up with. And so that's done for both the backbone main chain atoms and also the side chain atoms. In the side chain atoms, sometimes it's more than 1,4 interactions. These are known as, so Dunbrack comes from the Dunbrack rotamer library, which is just going into the PDB, looking at all the rotamer shapes for each amino acid type, noticing that each type of amino acid favors, you can cluster all of the side chains by their shape, and notice that individual side chain types tend to fall into one of a set number of shapes. And you can just bin those and say, okay, leucine only likes to be one of these set number of basic geometries, even though, in Rosetta you could represent that by, any floating point number. A more interesting thing is that you can notice that if you go into the PDB, for any given amino acid type, if you look just at the backbone angle, say leucine only likes to be one of 3 types of rotamer shapes when it's in a helix. And those distribution shapes change in the backbone. And if we violate those propensities, we get an increase in this term, the Dunbrack term. All the exact same thing is true for the Ramachandran plot. Phi psi angles, so different amino acid types like to be in certain phi psi bins. Glycine can do whatever it wants. Proline can only do a few things because it's limited. And so this is side chain. Both of these are backbone. This just is a spring that forces your backbone omega angle to be in what we consider good. And this is just kind of an add-on term to force our proline backbones to be where they're supposed to be. Yeah, that's all I need to say about that. And then just tacked this on it. Forgot to add. Very last thing is the reference energy. Oh, yeah.
So the Dunbrack score depending on
It's dependent on the backbone angles.
where the backbone angles come
No, they're not. When doing the derivatives in the code, yes. Like if you're keeping the rotamer fixed and changing the backbone angle, then it needs to go backwards like that. But not in the calculation really. You just, for a given phi psi bin, you changes the probability distribution of the Dunbrack term, but not vice versa, I don't think.
The they are both backbone and backbone Dunbrack libraries. There is some regions of sidechain confirmation states which are poorly represented in the PDB. Rosetta falls back in the backbone independent library if it doesn't have enough data for the backbone independent library.
And then, so the last one is reference energy. This only matters if you're doing design. For structure prediction, this doesn't change. And all this is it's a single value for each residue type. So if, say, like the reference energy for tyrosine is 0.5, and you have 10 tyrosines in your protein, doesn't matter where they are, then then that will add 5 to your score, right? And so the reason this exists is because, say you've got a helix and you're running the design simulation on it, and you're like, should I put alanine there or should I put phenylalanine there? If it fits, phenylalanine has way more atoms and more chances to have interatomic interactions. So without any reference energy, Rosetta is just going to put phenylalanines everywhere. It's going to put whatever residue has the most atoms everywhere because it can get better score. The thing it's not accounting for is that that every atom in that residue could also get those energies in the unfolded state over here, right? So in the unfolded state, phenylalanine is, say, interacting with a bunch of water, and, depending on what your religious beliefs about the unfolded state of proteins are, and other protein atoms, and it loses those when it gets into the folded protein. When we're just doing protein folding, the sequence is fixed. We don't have to account for that. It cancels out. But when we're doing design, we have to account for that. The way you'd like to believe this worked is we say take a whole bunch of fragments and try putting residues on all those to get an idea of what the unfolded state looks like, and then just average that out. But it doesn't actually work that way. What these actually are just 20 calibration factors, 20 fudge factors that we use for each residue type, and we just calibrate it by taking a large set of high-resolution protein structures, running all residue design on it, and then tweaking those 20 numbers to recover the native sequences the best we can. And we just assume that those are, for better or worse, those are the best numbers that we can come up with for the reference energies. And so one thing that, like, if you're getting too many, say you're getting too many glycines in this loop you're trying to design and you don't want that to happen, David might say, oh, well, just turn up the reference energy for glycine as kind of a hacky way to just make that not happen. That's what he's talking about. And that is all.
Quite often in Rosetta, we use Monte Carlo methods. And you'll hear this term over and over and over, and no one ever defines it for anyone. In general, Monte Carlo methods are methods for numerically approximating something that involve a random element. So you're going to randomly sample something, and then you're going to apply some rules to your sampling to extract out useful information. More specifically, most of the time when we're doing Monte Carlo sampling, we really mean a specific type of Monte Carlo sampling called Metropolis Hastings sampling. And the idea with Metropolis Hastings sampling is, I'm going to start somewhere. This could be with a known structure. This could be with a randomly generated structure. And I'm going to score the energy of that point using the score function that Chris just described. And then I'm going to make a random move. I'm going to take some sort of step in conformation space. And at the new position in conformation space, I'm going to evaluate the energy. So that's the random element. I've made a random move. And now I know the energy at my new position and my old position. If the energy has gone down, I accept the move and I stay where I am. If the energy has gone up, I randomly accept or reject the move with a conditional probability that depends on how much the energy has gone up. If it's gone up a lot, I always reject the move. If it's gone up a little bit, I might be very, very likely to accept the move. So if I take a step and the energy's gone way up, I step back to where I was, and now I make another random move. And I do this many, many times. So I do a trajectory in conformation space, taking steps which on average are going to take me down to lower energies, but which can go uphill a little bit to climb up out of local minima. This turns out to be a pretty good way of sampling rugged landscapes in higher dimensional spaces. And it's at the core of many of the algorithms that we use. Now, there's one other thing that I should mention here. When I take this step, and if the energy has gone up, and it's gone up by a medium amount, I need a way of calculating the probability with which I should accept or reject this move. And there's actually a factor called a temperature factor that helps to determine that. If the temperature is very high, I'm very, very likely to very likely to accept moves that increase the energy. If the temperature is very, very low, I'm very likely to reject moves that increase the energy. And a move that increases the energy would have to be very, very small for me to accept it. So the temperature lets you control how strongly you want to drill down into lower energy regions, or how much you're going to permit traveling uphill. So with a basic Metropolis Hastings search, you use just a fixed temperature value that's chosen for whatever task you're doing. But you don't have to do that. There's a somewhat more complicated algorithm called simulated annealing, where the temperature varies over the course of the trajectory. Usually at the beginning, you want the temperature to be very high so that you're accepting a lot of uphill moves. And gradually over time, you let the temperature drift down. The temperature sometimes can also vary depending on how many moves you've accepted or rejected recently. For instance, if I've taken a bunch of steps and they've all been uphill and I've rejected all of them, I might say, oh, okay, I must be in a local energy minimum, so I've got to raise the temperature and start accepting a few of these uphill moves so that I can start going downhill again. So that's what we generally mean by Monte Carlo methods. Does anyone have any questions about this? You look like you might have a question. That's a good question. Yeah, when do you stop? Stopping conditions can be arbitrary. So in some cases, you might want to stop when your energy has improved by a certain amount. In other cases, you might say, I just want to do a fixed number of moves, and then I'll stop. I think most of the time we're actually just doing a fixed number of moves and then stopping. In most cases. The other thing is, when you stop, you often have the choice of either just accepting wherever you've ended up or picking the lowest energy state that you've encountered on your search. And most of the time you'll want to pick the lowest energy state that you've encountered along. The question was, can we estimate what the values should be for the folded state so that we know whether or not we've found a global minimum? In practice, that's very, very difficult. The one thing we can say is that a folded state in Rosetta should probably have negative energy, but beyond that, we can't say a lot. The other thing we can say is if a protein folds, there should be a fairly large energy gap. So if we have an obviously unfolded state and a candidate folded state that's very close in energy, either that's a protein that probably doesn't fold or we haven't found the folded state. But yeah, it is very, very hard to know a priori what the target energy should be. Okay, so a common algorithm that gets mentioned over and over is fragment insertion. This is one of the things that's at the core of Rosetta's structure prediction by the ab initio structure prediction. It's also used for design in some cases. You build up desired scaffolds out of pieces of known proteins. And the basic idea is I want to search conformation space. Conformation space is too vast for me to search exhaustively. So I'm going to bias my search really, really heavily. And I'm going to bias it based on all the structures of known proteins that are out there. So my moves are going to be, I'll start with an extended chain, let's say. And I'll make a list of all these little pieces of proteins from the PDB. 9 residue fragments and 3 residue fragments are what are usually used. And I'll make this list based on the sequence of my protein. I'll take the sequence and scan along 9 residue windows and actually just make a list for each of those windows. And then what I'm going to do is I'm going to do a Monte Carlo type search where my moves are taking a random chunk of my protein, a random 9 residue window, and setting all of the backbone phi and psi and omega angles to the values from that fragment from the PDB. So you set a chunk to be like a known structure, and then you set another chunk to be like a known structure, and then you set another chunk to be like a known structure. And at every move, you can accept or reject that move based on what it does to the energy. That's the basic idea. There are complexities that I'm not going to talk about. Okay, the Packer. The Packer is a core algorithm in Rosetta which doesn't modify your backbone conformation at all. It only operates on side chains. So that's an important thing to remember. And what it does is it solves a very complicated very complicated combinatorial problem. Let's say you've got a bunch of positions with side chains, and you've got a bunch of different possible rotamers at each of those positions. If you had n positions and you had m possible rotamers at each position, you'd have m to the power of n different pairwise combinations of all those rotamers, which is a number that explodes as the number of residues goes up. It doesn't scale in polynomial time. It's a hard computational problem. So we can't tackle this exhaustively. We can't consider every possible combination of rotamers. So we have to do this using Monte Carlo methods. And the Packer uses a simulated annealing-based search. And what it does is it starts with your rotamers in some sort of conformation and calculates the energy of the structure. And then it takes a single residue and changes the rotamer. And it calculates the energy of the new structure. The new energy calculation is actually very, very, very efficient because, as Chris mentioned, everything in Rosetta is pairwise decomposable on the residue level. So when I change this rotamer, I don't have to recalculate the interaction of this residue with this residue, because that's going to stay the same. All I have to recalculate are the internal energy terms for this residue. So the Dunn-Brach term, for example, of Fa Dunn, which gives you an energy based on the current rotamer, and the pairwise terms between this residue and its neighbors. So is this residue now clashing with this? Is it now clashing with this? Does it now have a favorable electrostatic interaction? And I only update those energy terms for that residue and its neighbors. The other thing that's worth keeping in mind is a rotameric substitution doesn't have to be between, it doesn't have to preserve the amino acid identity. There's no rule saying it has to. So that arginine, for example, that I've got in blue there could be replaced by a tyrosine in one of these moves. By default, the default behavior of the Packer is to repack every single position in the protein and to allow design while it's repacking. So the rotamers it'll try are all of the rotamers for every possible amino acid type. And it can change the amino acid identities as it does so. Now it does this search, replacing amino acid identities, calculating an energy, and then accepting or rejecting the energy based on the Metropolis criterion. And it does a simulated annealing-based search. So it starts with the temperature very, very high. It'll accept all sorts of bad insertions that'll create clashes. And then the temperature drifts downward. So now the moves start to have to clear those clashes. So here I've got two tyrosines that are clashing, and eventually you end up finding a set of rotamers that are pretty good. Limitations of the Packer are, one, it doesn't move the backbone. Two, if you give it every residue and let it design at every position, it's got a huge number of combinations to search through. So it's not guaranteed to find the best possible solution. It'll find a solution. Three, it's stochastic. It's random. So if you run it a bunch of times, there's no guarantee you'll get the same solution every time. And four, it doesn't do any off-rotamer optimization. So it's sampling rotamers discretely, but you'll never sample something that's halfway between. The behavior of the Packer is controlled by task operations. You've seen these in Rosetta scripts. Task operations never affect the backbone. They only affect side chains. They only control the Packer. And it's very important, if you don't want to design your protein, for example, if you're just relaxing it, to give it a task operation that says, don't design. Preserve the amino acid identity, so that now, instead of considering every possible side chain and every possible rotamer at this position, I'm only going to consider tyrosine if this is a tyrosine. Task operations are multiplicative. They can only say, don't do this. By default, the Packer does everything it can. It designs and packs. A task operation can say, don't design. So if you say don't design, the Packer will just pack. It can say don't pack, in which case at that position the Packer will just preserve the native rotamer and it'll only do rotameric trials at other positions. But if I say don't pack at this position, there's no way that I can come back and subsequently say, okay, pack. if I say don't pack at position A and I say don't pack at position B, the packer is going to say, okay, I can't pack at position A or position B. So does that make sense? But the packer is only, or task operations are only prohibitive. So that's the packer. That's for optimizing sidechain placements or sidechain identities. And that's what is usually used for design. There are other algorithms as well. The minimizer, this gets back to what you asked about how flat the energy landscape is. Let's say I'm at some point in the energy landscape, and rather than doing a broad search to find a deep well that might be far from my starting point, I want to do a narrow search where I just roll downhill and find the nearest local minimum. If I were up in the mountains and I just wanted to find the nearest local minimum, I would just walk downhill until I find the nearest local minimum. And that's pretty much exactly what the minimizer does. Warning, there's a little bit of math on this slide. So this point in confirmation space, remember, can be represented as a vector of numbers. We can calculate what's called the gradient. The gradient is and actually I'm calculating the negative of the gradient. The gradient is a vector of derivatives. And here I'm calculating the derivatives of the energy function with respect to each of the degrees of freedom of the system. The gradient of a function in a certain space points uphill. If I take a step in the direction of the gradient, I'm going to be increasing the value of that function by as much as I possibly can. So the negative of the gradient points downhill. If I take a step in that direction, I'm going to be decreasing the function by as much as I possibly can. So the basic algorithm for the minimizer is calculate start at a particular point, calculate the gradient at that point with respect to the degrees of freedom of the system. So if we're working in dihedral space, we calculate the gradient of the energy with respect to all of the dihedrals that you can rotate in the protein. Take a step in that direction, which means moving all those dihedrals by a small amount, and actually do what's called a line search. Keep stepping in that direction as long as the energy is going down. When it starts going up again, you know that it's time to calculate the gradient again. You might not have found the local minimum yet, but you've at least found a point where you need to adjust for the fact that the path downwards might curve. And so do it again. Calculate the gradient again, change your direction, start walking downhill again until you start walking uphill, go back to where you were at your lowest point, calculate the gradient again, and keep doing this until you get to a point where the gradient is very, very flat. And that's where we consider how flat the energy function is. The minimizer has parameters that you can set, including a threshold value that determines how flat the gradient has to be before you stop searching, before you say, okay, I'm going to accept this as my local minimum. Limitations of the minimizer: it can only find a local minimum. It can't find a global minimum. There's another important caveat as well. It's very inefficient to do just gradient descent because, as I said, the path downwards might curve. And that means you would have to do a very short line search, calculate the gradient again, do another very short line search, calculate the gradient again, do another very short line search, which is inefficient. The way to make it better is to use information about the curvature of the surface as well. That means second derivative information. To do that, that would involve calculating this, which is called the Hessian matrix. It's a matrix of second derivatives. The matrix of second derivatives scales with the square of the number of degrees of freedom in your system, which is not nice. It's computationally expensive to calculate it. It takes a lot of memory to store it. So we don't want to do that. Instead, lots of mathematicians have come up with approximations for the Hessian matrix that just use the gradient from the last several steps. If I know that the gradient was like this before, and now it's like this, and now it's like this, I can say something about how the surface is curving, and I can anticipate that it'll curve even more this way. So, and if I know that the surface is curving this way, rather than taking tiny steps like this, I can probably take bigger steps in the direction that it's curving. The minimizer has options for how this approximation is made. The default is DFP-min, Armijo, non-monotone. There are all these alternative ones. I'm not going to get into the details of these, but basically these are approximations for the second derivative matrix. And you can play with these if one of them is not giving you the behavior that you want. The LBFGS one was added more recently. I think it is a little bit faster than the default, the DFP-min one. So that's the minimizer. Any questions about it? Mm-hmm.
How is it different from Relax?
Relax uses the minimizer. Or the current versions of Relax use the minimizer. In fact, I'm going to talk next about how the fast relax algorithm works. Okay, all good? Okay, so more complicated algorithms in Rosetta are by and large built up out of the simple ones. So as an example, here's the fast relax algorithm, which contrary to its name is not fast, because it's built up out of all these smaller steps which are fast. With fast relax, we say, okay, first I'm going to set the FA repulsive term to a very low value. What this does is effectively smooths out the energy landscape a little bit, so that some of the local minima that were blocking us before are much smaller, and rolling downhill is likely to take us further towards the global minimum. So I set the repulsive term to 2% of its normal value. Now I call the packer. The packer is going to find a solution which might introduce some clashes because the repulsive term is so low. And then I'm going to call the minimizer with relatively low stringency. And it'll relieve some of those clashes a little bit, but because the repulsive term is so low, it's not going to do much to relieve the clashes. But it could go quite a ways because clashes are not blocking the protein from moving at this point. And then I'm going to ramp up the repulsive term, and I'm going to call the packer again, and I'm going to call the minimizer again. And then I'm going to ramp up the repulsive term some more, and I'm going to call the packer again and call the minimizer again. And then I'm going to set the repulsive term to its normal value, and I'm going to call the packer for a final packing, and I'm going to call the minimizer, and this time I'm going to set the minimization threshold very, very low, so that this is a very high stringency minimization. And it gets you to deeper minima near where you started a little bit faster. Fast relax, by the way, you should give it a task operation that says don't design. There is also a fast design algorithm that works similarly but allows design during the packer steps. The one thing I forgot to mention, the minimizer can operate on different degrees of freedom of your system. If you want, you can tell it only minimize this little stretch of the backbone over here, which means that it's only going to be calculating derivative vectors with respect to those degrees of freedom. The degrees of freedom that the minimizer operates on are governed by move maps. And the minimizer, unlike the packer, can work on both side chains and backbone, or just side chains, or just backbone, or whatever you want. So move maps control the minimizer, packer, task operations control the packer. And that is pretty much it. Are there any questions? Mm-hmm. Yeah, you've probably seen these in Rosetta scripts. Move maps basically just tell you, with a move map you specify what regions of backbone are fixed, what regions of backbone can move, which side chains are fixed, which side chains can move, and which jumps are fixed and which jumps can move. Jumps are also things that the minimizer can modify. Move maps, unlike task operations, are not just prohibitive. If you say in a move map, don't allow any of the backbone to move, and then you subsequently say, do allow these residues, backbones, degrees of freedom to move, it's whatever you say last that takes priority. So you can turn things off and you can turn things on in a move map. In a task operation, you can't do that. If I've said nothing can pack, I can't subsequently say, oh yeah, you can design these positions. By the way, designing depends on being able to repack. If you prohibit repacking, you also prohibit designing. I think there are some things in the Packer that are smart enough to prune the list of allowed rotamers a little bit. So for example, if there were a position where that was glycine and where the C beta was always going to clash with the backbone, then there's no point in trying any rotamer that's not glycine, because no matter what I put in there, I'm going to have a clash with the backbone, and I can't move the backbone of the Packer. So I think there are some things in the Packer that do prune the rotamer list a little bit. Each fragment insertion move is followed by an energy evaluation. So if I've randomly picked position 41 to 49 for an insertion, I make an insertion there, and then I accept or reject the move before I make the next insertion, I think. In the Rosetta database, there's a section for all of the elements. I think Rocco recently has gone through and added the whole periodic table. There's also, so you'd have two separate considerations. One would be, can Rosetta work with a residue type that just has this atom type? And for that, I think Rocco now has added the whole periodic table. The other is, do I have a residue type in the database that has whatever atom? So if I want to just work, if I just want to work with a zinc, for example, there already is a residue type in the database, which is just a single zinc atom. Because of some of the weird internal workings of Rosetta, Rosetta doesn't like residues which are just one atom. So the zinc residue type is actually zinc plus 4 virtual atoms. Virtual atoms are atoms which are not scored, which are ignored by the score function. How do you alter backbone conformation? You have algorithms which alter backbone conformation include fragment insertion. The minimizer can locally alter backbone conformation. And then there are all sorts of specialized algorithms, including loop hash, kick, things like that, which can create local backbone perturbations. There's also the small mover, which creates a random local backbone perturbation. The shear mover, which creates a random local backbone perturbation, minimizing the effects upstream and downstream, and so on and so forth. So that the short answer is there are a bunch of movers which modify backbone conformation. One I should have mentioned earlier, one of the core paradigms in Rosetta is you've got a structure, you're going to do stuff to it. The stuff you do to it is done using things called movers. The Packer is one type of mover, the minimizer is one type of mover, the fragment inserter is one type of mover, loop hash is a mover, kinematic closure is a mover, etc. So a mover is something that takes a structure, does something to it, and spits out a structure. Changes the sequence, moves the backbone, does something. Thank you.